Microbial fuel cells. Principles of MFC. MFC is a bioelectrochemical system that converts bioconvertible chemical compounds to electricity under the catalytic activity of microorganisms. Very recently, these MFCs attract the attention of researchers and became promising for practical applications for treating wastewater. In MFC, biomass is first converted to substrate fuels such as glucose, xylose, acetates, which are utilized as carbon sources by microorganisms. A typical MFC consists of an anode, a cathode, a proton or cation exchange membrane and an external electric circuit. In the anode chamber, the microorganisms convert the fuel into carbon dioxide, protons and electrons. The electrons are transferred to an insoluble electron acceptor, the anode, via a redox mediator and then flow through an electrical circuit with a load to the cathode. Thus, electric power is generated by the potential difference between the anode and the cathode. In the anode chamber, the protons flow through proton exchange membrane to the cathode, where they combine with oxygen and electron to form water. A catalyst at the cathode or a catholytic solution is used to facilitate this reaction. The byproducts of the reaction are carbon dioxide from decomposition of organic matter and a small amount of water at the cathode. The microorganism is the bridge linking between chemical energy of the substrate compounds and electric energy. They obtain energy to support their life by transferring electrons produced from the oxidation of organic compounds to the metabolic products and finally to CO2 in MFC. Thermodynamically, this energy comes from the change of free energy delta G for oxidation of the substrates by exogenous oxidants. Respiration and fermentation are two identified major metabolic pathways for electron transfer. Using glucose as an organic substrate, 24 electrons and 24 protons are released in the anode chamber. These protons and electrons both travel to the cathode chamber, which combines with 6 molecules of oxygen to produce 12 molecules of water and 6 molecules of carbon dioxide at anode. Direct electron transfer. Electron transfer can occur through direct contact between the microorganisms and the electrode or with the use of nanowires. Direct contact requires that the organisms have membrane bound electron transport protein relay such as C-type cytochromes to facilitate the transfer of electrons out of the cell. However, this transfer allows only for one layer of bacteria in direct contact with the electrode to transfer electrons. Nanowires are conductive appendages that are hypothesized to carry electrons from the bacterial cell to the surface of the anode. The bacteria can attach to the anode of the MFC and form biofilm, which is a multi-layer community of bacteria that can theoretically grow to any thickness. The nanowires connect cells of the outer layers of the biofilm to the inside of the system and allow the electron transfer to the anode. This can allow thicker biofilms to develop, which provides increased current production from the MFC. The electrochemical detection of redox signals demonstrate that some domains of the outermost microbial surface are close enough to the electrode to allow electron transfer. Mass transfer and kinetics. The bacteria that are transferring electrons exocellularly can attach and grow on the electrode, forming a biofilm. These biofilms are made up of numerous organisms and can theoretically grow to any thickness. As these biofilms become thicker, the mass transfer of nutrient and substrates to the microorganisms attached to the electrodes is facilitated. Electrochemical analysis of MFCs. Through electrochemical analysis of MFCs, we can obtain experimental results by performing electrochemical redox reactions in MFC. Typically, anode, cathode and reference electrode were connected to a potentiostat using a copper wires and 1000 ohms as an external resistance. Current output density is measured and plotted against the time. Similarly, CV studies are used to analyze the electrocatalytic behavior and interaction between the redox mediator, biofilm and anode. The CV data is recorded using 3 electrode system where anode, cathode and silver chloride electrode were considered as working, counter and reference electrodes respectively. The CV in the presence and absence of biofilm are determined at various scan rate with potential range between 1 to -1 volt using automated electrochemical workstation. From the CV data, Coulombic efficiency and power density can be calculated. Voltage and power generation. Generally, electrochemical analysis of MFC results are discussed in terms of Coulombic efficiency and power density. Let us see how it is obtained theoretically from electrochemical reactions in MFCs. Voltage generation in a microbial fuel cell can be obtained by E is equal to IR, where I is current and R is external resistance. Electricity can be generated in a microbial fuel cell if the overall reaction is thermodynamically favorable. The thermodynamic property Gibbs free energy can be used to determine the maximum amount of work that can be obtained from the reaction by delta G is equal to delta G naught plus RT ln Q, where delta G is Gibbs free energy for specific condition, delta G naught is Gibbs free energy for standard conditions, R is the universal gas constant, T is the temperature and Q is the reaction quotient of the activities of the product divided by the activities of the reactants. The cell voltage, also known as electromotive force, can be related to free energy as E naught is equal to minus delta G naught by NF, where E naught is the standard electromotive force, N is the number of electrons per mole of the reaction. EMF can be calculated for anode and cathode using E is equal to E naught minus RT by NF ln Q. Thus, total cell potential can be calculated using half cell reactions occurring at anode and the cathode of the microbial fuel cell as E is equal to E cathode minus E anode. This equation can only be used in anode and cathode reaction running at the same pH. Once we obtain cell potential with the known external resistance, power of EMF can be calculated as P is equal to E square by R, where E is equal to voltage recorded from microbial fuel cell and R is the external resistance. When reporting the results, Coulombic efficiency of the system is important parameter to describe the overall efficiency of the MFC. Coulombic efficiency is the fraction of electrons that are recovered as current versus the electrons that were in the starting substrate. It can be obtained as C is equal to M integral I dt by F B delta C and V, where M is the molecular weight of the substrate, I is the current, F is the Faraday constant, B is the number of moles of electrons removed through the oxidation, V is the volume of anode compartment liquid and delta C is the substrate concentration change over the batch cycle.